Our study attempts to bridge an outstanding gap in the field of aging, a major risk factor for chronic diseases, and a multi-system problem as we age, drawing on multiple organs and tissues and unhealthy aging patterns. Human aging has recently been typified into 4 separate ageotypes associated with renal, kidney, hepatic, liver, metabolic adipose tissue, and immune dysfunction. What is intriguing is that with immune dysfunction, you get the increased propensity for the involvement of all the other 3 tissues and unhealthy aging patterns. Immune dysfunction as a hallmark of aging is generally typified by global dysregulation of central recirculating components and how they shape tissue function. So B cell, T cell, NK cell function, and how they change the outlook for pathological aging. The same can be said for innate immunity and aging and the relationship therein. Most of what is known and the gaps are shaped by the context of recirculating innate immune effectors like neutrophils and monocytes and NK cells and how they shape various tissue function and pathology with age. The premise for our work is, of course, the fact that inflammation is a potential driver of aging-associated dysfunction in chronic diseases. However, we're specifically focused on the role of tissue-autonomous innate immune responses in aging, which has largely been ignored in the context of aging, and it is also to define a hallmark of tissue inflammation with age, which has also remained outstanding. In order to do this, we have comprehensively aged out mice to different time points, collected at least 6 separate tissues in a synchronized manner, subjected them to 3 separate overlapping techniques: single-cell RNA sequencing, high-throughput immunophenotyping, functional and histological examination, to tie in cell and tissue-specific immune signatures and distribution through single-cell RNA sequencing, and corroborate those patterns with immunophenotyping, and draw out any relevance and physiological components with functional histological examination. The major conclusions from our work, as you will note, and multiple points of novelty, include that tissue autonomous inflammation is a novel hallmark of aging as we have defined it. Intriguingly, aging tissues possess common but also highly unique modules of inflammatory signatures that define their functional states. A core component of this unique signature of inflammation are type 1 interferon derived gene signatures that characterize aging tissues and can be considered a novel hallmark of aging tissues. These ISG signatures predominate in the stroma, which is surprising, as up till now, much of immune dysfunction has been correlated to immune effector cells. Aging dramatically reshapes tissue-associated myeloid compartments and specifically leads to the loss of embryonic tissue-resident macrophages, a core component that interacts intimately with the stroma. We further define the role of the ER-resident stimulator of interferon genes, which senses cytosolic DNA and shapes type 1 interferon signatures downstream in aging stromal subsets. We have also found that STING signaling, it puts the brake on embryonic tissue-resident macrophage loss with age, which is an unexpected and paradigm-shifting observation. Overall, our different ways of corroborating the phenotypes have led to the recapitulation of heterogeneity in all these shifts by immunophenotyping, and we are now tying this all into relevant physiological outcomes. Thank you.